I don't have a page. Yeah. Yeah. So we see put it in. We didn't do it before. Yeah. Okay. Sounds good. Oh no. What are you gonna do on some okay. Yeah, I understand. Like, I understand. Yeah, because like I understand the second. I understand how to do the lab, like doing finding. Yeah, I just didn't understand the first one. Because that's so fast. Yeah, but then the other problem we did, like, yeah, when we use the contract. Oh. Yeah. I understand how to get like figure out which one is the higher cost.
Dan, can you hear me?
Yeah, I can.
Turn your microphone on and say something. Can you hear me?
Yeah, can you hear me? Can you hear me?
Okay, folks, let's get started. So we have somebody who's sharing. We need to Zoom today. The men's soccer team, apparently there is potential exposure. So our dear soccer player, he's not tested positive, but he apparently was potentially exposed to somebody who is potentially positive or something like that. I have no idea, but he's been advised not to come to class until further notice. Your mother-in-law's former roommate. Yeah, her dog or something. Okay, let's go. We're starting. Hello. So today we're going to start with amino acids. We've already dealt with these in lab, but now you can learn about them just formally. Let's have a word of prayer, then let's dive in. Father, thank you. We get to learn. Thank you. We get to learn about how we, and in fact, your creation is in fact wonderfully made. And we thank you for the chance to learn, even under not very ideal circumstances. Guide us now in Jesus' name. Amen. Come on, you. Okay. There we are. No, not coming up. Yes, no. All right, let's stop sharing. Try it again. Share. Okay, good. And now. Oh, good gravy. Well, technology, guys, got to try and figure it out. There we go again. There we go. Okay, as a rule, by weight, definitely, proteins are definitely the most abundant biological macromolecules on the earth. That's, I think, pretty evident when you take a look at their diversity, when you take a look at what percentage of an organism is by what percentage of the mass is due to proteins. Proteins are easily the most abundant class of biological macromolecule really on the face of the earth. So proteins are polymers. Their polymers are linear polymers of these 20 different amino acids. You link those 20 different amino acids in various ways. You get wide variety of sequences, wide variety of combinations, wide variety of structure and functions and things like that. And the subunit of a protein, of course, is amino acid. So proteins are structural in nature. Think, for example, about bone. Let's think of bone. So bone has a organic matrix. That organic matrix is then a foundation upon which you precipitate calcium hydroxide salts. Without the biomolecules, without the protein, the bone-specific collagens, the osteopontin, the osteocalcin, the osteo-whatevers, all of those essentially make up bone. The cells scattered, but bone, besides the inorganic parts, are largely protein, believe it or not. Think about a tendon. Tendons do have a resident fibroblast population called the tenocyte, but a tendon is largely collagen, mostly protein. You think about the structures between cells, holding cells together. Actin is glue for a cell. That's made of protein. You go inside the cell, most of your macromolecular assemblages are made of proteins. So structurally, proteins are huge. They really are huge. They act as enzymes. The clear majority of all enzymes are proteins. So these are your biological catalysts that will increase the rate of reactions so that they can occur in real time. There's signaling molecules. There are groups of proteins called growth factors that tell cells to grow. There are groups of other proteins that tell cells to die. There are groups of other proteins that tell cells to move this way, move that way, turn this gene up, turn this gene down. Don't move. Go this way, don't go this way, so on and so forth. They are signalers also to cells. Transporters are embedded in membranes. They allow the means by which things pass through membranes. They're pores, they're active transporters, they're facilitated diffusion transporters. It's pretty fair to say, I think, not many things get across the membrane without protein transporters. They also regulate the expression of genes. So all of your polymerases are proteins. All of your DNA binding proteins are proteins. All of your things that regulate transcription, repressors, things like that, they're all proteins. Chromatin is at least by weight 1/3 protein. No, 2/3 protein when you include non-histone proteins. So that's 67% of the weight of chromatin. So gene expression is also intimately tied in with proteins. The function of a protein is going to be a consequence of its structure. So when we look at protein structure, that's going to be a very important distinctive, and that distinctive will play a role in its function. And the amino acid sequence of the protein is going to, in many ways, determine its structure. And so here's my kind of take-home lesson from this slide. Structure of the protein is going to largely determine its function. You could probably state this the other way, that the function of the protein largely arises from its structure. So structure and function of the protein are intimately associated, they're inextricably linked. So, yeah, Amanda?
Earlier in the semester, you had mentioned that something else determines the structure of protein that's not the amino acids. What is that?
The way the protein is made have a lot to do with the structure because some, for example, insulin. Insulin, your favorite protein. Insulin is made as a precursor and then it's processed multiple times. And the structure that it has, it would not have if you just threw insulin in sesame said fold. It folds as it's not pre-protein, it's a pre-pro-protein. And the pre-pro-protein folds and then it's processed, and then that's what you get out of it as well. So sometimes the structure it takes in its precursor form is what will determine what it actually is structurally final. Also, you can modify the structure of proteins by covalently modifying amino acids. You get a bulky amino acid, that changes its structure. Also, the binding of other proteins to it can change its structure. There's actually a group of proteins that are called unstructured proteins. They're not folded into any coherent structure, 3D structure. In some cases, they assume a structure when something interacts with them. In other cases, they're just unfolded and they function that way. So that lack of structure is actually an important part of their ability to function. So there are other things that can influence it. The most important factor when it comes to the structure of a protein is the amino acid sequence of the protein, by far, easily, by far. So in nature, there are 20 amino acids that occur naturally. There are plenty others that are synthetic, but the 20 naturally occurring ones are the ones you'll be responsible for. These are all alpha amino acids. Who can tell me what an alpha amino acid is? Right. Alpha carbon, that means it's the first one away from the carboxylic acid, is a chiral carbon. The amino acid, the amino group, the main group is attached to the alpha carbon. Attached to the alpha carbon. All of your 20 amino acids also have a carboxyl group that's attached to that same alpha carbon. So they're all going to have this generalized structure like this. Carboxylic acid, hydrogen, and whatever. And the whatever is what's going to determine the chemical properties of this molecule. And to remember the structure of these, just draw this out. This is simple enough to remember. And all you have to remember really are the whatevers. And then you can match the whatever to whatever amino acid you're trying to get to. So the identity of the amino acid depends on the structure of that R group, the functional group it is called. The functional group will vary in terms of size and charge and solubility, hydrophobicity, hydrophilicity, things like that. And then you're going to count carbons, designating them as beta, gamma, delta, epsilon, so on and so forth. Counting away from the alpha carbon, which is the carbocarbon. So here's an example. Here's lysine. Here's your alpha carbon. And then you go beta, gamma, delta, epsilon, and you have an epsilon amino group. That's essentially how you get lysine. So 1, 2, 3, 4 amino group, that's your lysine right there. Now you can use also numbers. If you do numbers, you start with the carboxyl group. 1, 2, 3, 4, 5, 6. Carbon number 6 or epsilon carbon. Either one, I will accept either one. Both are found in the literature, both are equally used. Jenna? Yep. That's your alpha carbon. Yep, we do. So carboxyl is always C1, carbonyl C2. And if you're going alpha, it's always, alpha is always the carbocarbon. The other R group carbons will be designated after the carbocarbon according to their distance from that carbocarbon. So amino acids can exist in enantiomeric forms. So here we have one whose R group is a methyl group. This is alanine. So alanine, as we see here, can exist as enantiomers. Let me hold these a little closer. Okay, Dane, buddy, here you go. Enantiomers. Oh, can you say these are the images of each other? Now, let's determine these stereoisomers. So, who's one, who's two, who's three? Show me one. This is element number 6, element number 7. Okay, show me number 2. Okay, and of course, number 3, you get booty balls. Okay, so you hold it away, you go like that, that's the way the clock goes. So that's the right way, that is R. Okay, and point number 1. No, that's not
Oh, sorry, I'm going back.
Yeah. Okay, Brent, that's number 1, show number 2. Okay, and then Rich is number 3. Okay, so you want to please, I'll pull that way. Oh, stupid, that's stupid way. Yes. All right, so this is R. This is S. Yeah. And biology. S is going to predominate. S will predominate. L. The amino acids you often have L and D. L is S, D is R. So Dan, if you didn't hear that, the S is a stereoisomer that tends to predominate in living systems. And also the S and the R are often called L and D. L and D. Yeah. L is S. And the way I remember that is L left. S in Latin is sinister, and L left, sinister, L and S. They go together. D is dextro. Dextro in Latin means right hand. So R, right, dextro, D, and letter. Oh, there is one amino acid whose R group is an H. Since it has 2 H's, can't be chiral, can it? That's glycine. So glycine does not exist in stereoisomers. All the others do. All the others do. And oh, look, gee, there's lysine. And lysine, as you can see, has a methyl group, has a tar group, and it exists in the 2 stereoisomers. And we figure that out here. I'll leave it to you essentially to convince yourself that this is the way these go. These are, in fact, mirror images of each other, pretty obviously. So alanine can exist as L, can exist as D. This is a form you tend to find in living systems. And as we said, yeah, I said it there. Now we're going to see this actually in the lab. We're going to do, not tomorrow, but a week from tomorrow. Because your biological machinery is rigged to handle S stereoisomers, L stereoisomers. If you give it a D stereoisomer, the enzyme kind of looks at it and goes, what do I do with that? Virtually your whole biology is geared towards using the L stereoisomer. And we're going to see this, we're going to give an enzyme a stereoisomer of its substrate, and the enzyme is going to go, huh, what do I do with this? Yeah, usually. It's hard to isolate stereoisomers sometimes, so they'll just give you the racemic mixture most of the time. Then figure out a way to isolate stereoisomer, then mark it under another name and get another patent. And extend the patent price and make more money off it. Cool, huh? Yeah. Yeah. Sugars are found in the D. Yeah. It's opposite with sugars. Yep.
The D-stereoisomers when you take like a racemic mixture of medication.
The Ds will tend to.
So like, they're racemic to dilute the L stereoisomer, or just.
They just can't isolate them. Isolating them can be very difficult and extremely expensive. That's the other thing. So if you take, say, I was reading the other day. So there's this company that sells this mixture of the 9 different essential amino acids, but they make them synthetically in the lab. So they make them into a synthetic mixture. So I was looking at this and I was saying, these are synthetics. I bet you there was in mixtures. So probably close to half of what's in there is something your body doesn't use. So I wrote to the lab and I said, the PR people, and I said, these racemic mixtures of the amino acids. They said, well, I need to talk to our lab people. We'll get back to you. And then they got back to me and said, thank you for your interest. This contains a mixture of all 9 amino acids that you need. And it really works. And here's these testimonials for people who didn't answer. Which tells you the answer is yes. So you're paying essentially for half of stuff that doesn't do anything. Now, do you find D isomers in biology? Yes, you do. You find them in the cell walls of bacteria. So those D stereoisomers are the exception to the rule. Now, where do you think bacteria cross them? Where do bacteria make them? Bacteria make them from the L stereoisomer. We have enzymes called racemases. Racemases. And those racemases take the L stereoisomer and convert it into the D stereoisomer for use in bacterial cell walls. Bacteria probably do this to confuse the machinery of other organisms from degrading their cell wall. Well, not really, because they're pretty small, those amino acids. Excrete them. Yeah, pretty much. The amino acids are classified according to their R groups. Some of those are nonpolar. And the nonpolar ones include glycine. Glycine is the easiest. All of you should be able to remember glycine easily. Glycine, single letter code is G. Alanine, which has a methyl group. Single letter code is A. Valine, which has essentially an isopropane. Its single letter code is V. Leucine, its single letter code is L. Isoleucine, single letter code is I. These both have butane groups, but it's where the butane group attaches to the carbon that matters. Methionine, or M, which is sulfur-containing amino acid, one of two, and proline. Proline's a weirdo. You'll see why in just a bit. So, how do you learn these? Here's my advice. Take scratch paper. I always found the best place to get scratch paper is in the library next to the printer. People hard stuff that they mistakenly printed. Turn that scratch paper over and draw them and draw them and draw them and draw them. Draw them with the book, and then when I did this, when I took biochemistry, if I could draw them 5 times, not mistakes, not looking at the book, and I went on to the next one. And after I learned the next one, then I draw the other one that I learned, I draw them together. Make sure that I remember them. I go on to the third one. Oh my gosh, that takes forever. Yep. But you know what? I learned them.
So we should know how to draw all points.
Yep. Start together. Methionine, as you can see, has sulfur. Also has a methyl group here. If you donate that methyl group, then you form something called homocysteine. Homocysteine. And homocysteine is a molecule of interest because high levels of homocysteine in your blood tend to correlate with a high risk for atherosclerosis and coronary artery disease. Leucine and isoleucine, you can see, are similar. This one tends to have a 2-butane with the 2 attached to the alpha carbon. Here, leucine has an isobutane. And the number 1 carbon here is attached to the alpha. Carla?
What's the difference then from homocysteine and regular
Cysteine only has 1 methylene group and a sulfhydryl group. When you donate this, now this becomes a sulfhydryl group, except it has an extra methylene group. Valine, there's your propane, or 2-propane, if you will. Alanine, glycine, proline swings around and binds to itself. Proline is the weirdo. Proline has a ring essentially made of its own R group. And we'll talk more about proline because structurally, protein tends to put kinks in protein chains. So structurally, this is a very important amino acid. Uncharged amino acids. These are polar, but they're not charged. Polar, but not charged. Come on, you. There it goes. Yeah. So these include cysteine, which is the other sulfur-containing amino acid. Its singleton code is C. Serine, singleton code is S. Asparagine, now they get weird. Singleton code is N. Glutamine, singleton code is Q. And threonine. Threonine and serine contain alcohols. So serine has as its R group a methanol. Threonine has as its R group an ethanol. The hydroxyl group is significant because sometimes in proteins, carbohydrates will be attached to serines and threonines. Sometimes. And here's the structure of those amino acids. There's serine. Serine looks a lot like alanine, doesn't it? Threonine, here's your ethanol. Cysteine, you've got a sulfhydryl group. Cysteine is going to be really important for protein structure because of disulfide bond formation. Asparagine, glutamine, these look a lot like glutamate and aspartate. In fact, they're made from glutamate and aspartate. Then you have 3 aromatic amino acids. They all have groups with lots of resonance. These are very hydrophobic. And they can show aromatic stacking interactions. So they have pi orbitals and all that other sort of stuff. Obey Huckel's rule, blah, 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 all that. Phenylalanine, its singleton code is F. F. Weird. Tyrosine, single letter code is Y. Weirder still. Tryptophan, its single letter code is W. Don't look at me. I just report the news, I don't make it. Now, see this? Alanine, phenylalanine. You know alanine, you know phenylalanine. You know phenylalanine, you know alanine. Tyrosine looks exactly like phenylalanine, but you got that bloody alcohol here. So you know this one. This is the one that will challenge you probably. This is the toughest one to remember. This is called an indole ring. An indole ring. So you have a methylene group and an indole ring, and that is tryptophan. How do you spell it? Indole, I-N-D-O-L-E. And both of these rings are aromatic. This is very bulky amino acid. You have this tucked away in a protein, it's going to need room. And if you stick one of these in a protein where there's no room, the protein now is going to come unraveled because that bulky amino acid is shoving things aside to make room for itself. He has his cat on his lap and he's playing with him. It's very silly. Kitty. So these aromatic amino acids absorb at A280. Now, you can use A280 as an assay for protein because there is usually a certain percentage of the protein that will contain aromatic amino acids. The more they absorb at A280, that usually means the more protein you have. Did any of you have molecular genetics with Dr. Leonard? Did you do A260, A280 ratios? What does that tell you? Well, and also if your stuff is contaminated with protein.
Oh, yeah.
So if your 280 is really high,
Really? The difference between DNA and RNA? Or DNA and protein?
That's DNA and protein mainly. RNA, because of the uracil, will tend to absorb little, more like 250, 255. But 280 is usually, is at 260, 280, that tells you essentially how much protein is in the DNA. That's what that ratio tends to tell you. And you can use this as a quick and dirty way to detect protein. Here are your basic amino acids. There's 3 of them. These are positively charged at physiological pH. Lysine, single letter code K. Arginine, single letter code R. Histidine, single letter code H. So lysine, we looked at already. Here's a guanidino group, or arginine with 3 methylene groups. Histidine has a imidazole group. You use this in organic chemistry as a catalyst. So you know the structure of imidazole already. There's a methylene group on there, and zava boom, there you are. There's histidine. Histidine has a pKa very close to 7. So it can be protonated and deprotonated quite readily. So histidine is found in the active site of a lot of enzymes. It's also used if you want to change the structure of a protein subtly, because you can protonate that histidine or deprotonate it usually pretty easily. And then there are the negatively charged amino acids. These are acidic amino acids. There's 2 of them, aspartate and glutamate. The difference between these is 1 methylene group. Glutamate has the extra methylene group. Aspartate has only 1. So this essentially is acetic acid. This is propionic acid. Aspartate, glutamate, single letter code is D. Single letter code is E. D and E. Proline is a so-called secondary amino acid. A secondary amino acid. That secondary nature has to do with the fact that its R group carbons swing around and form a bond with the amine. So this is a so-called amino group, not an amino group, an amino group. So this makes proline moderately polar, also makes it really rigid structurally. And proteins that have stretches of prolines tend to be extremely inflexible, very rigid. And it's sometimes called an amino group with an I. Yeah. And the way you make it, you essentially make a glutamate, then you turn the glutamate into an aldehyde, then the aldehyde self-reacts like that, because aldehydes are really reactive and they swing around and react with that amino group, and that's how you make proline. You don't even have to, that's non-enzymatic even. You just have to take the carboxylic acid and you just have to reduce it to the aldehyde. All right. So cysteine, the sulfhydryl bonds, these can be oxidized. If you oxidize them, these are close enough together, you'll form cysteine. Cysteine. And these are important for a variety of structural considerations when it comes to the tertiary structure of proteins. We talked about this already with histidine, but it's found at the active site of lots of different enzymes. Contains an imidazole ring, pKa, about 6.5, very close to neutrality. Okay, here you go. They're responsible for the structures of all 20. They're also responsible for their single letter codes. I should be able to write a protein sequence, single letter codes on the board. You shouldn't have to ask that's what. You should know. Okay, so I'm going to make that expectation. How long will it take you to learn these? I don't know. I don't know. It depends how fast you memorize stuff. Maybe you'll learn them quickly. I don't know. But I do know that if you draw them out, you'll learn them faster than just by looking at them. Okay, Dane, hear me? All 20. Okay, yeah, good, okay. Tell the cat too, all 20. Start now. Start now. Don't say Thursday, oh man, I got a quiz Friday. Oh, let's see here. Better start learning these because one of them is going to be on the quiz. No, start now.
So we should have them memorized by Friday.
Yeah, you should. You should. Structures, as in able to draw them. Now, as we said before, the R group is what confers the chemical properties on the amino acid. So your aromatic amino acids, they're strong absorbers of UV light. They are also capable of hydrophobic stacking interactions. If you get a bunch of them together, they can swing their rings like this and stack. That can provide additional bonds within a protein to hold it together. Lysine, arginine, both have positively charged R groups. That's going to make them charged. And then you have particular amino acids that are non-standard. How do these arise? They arise post-translationally. Post-translationally. Dave, now post-translation, what does that mean? After translation, meaning after. Right, you're exactly right. After the protein is made, the amino acids will be modified. Let's take a look at some examples of non-standard amino acids. Here's one, 4-hydroxyproline. 4-hydroxyproline. 1, 2, 3, 4. 4-hydroxyproline. Why would you put a hydroxyl group on proline? Any guesses? Hydrogen bonding possibilities? Does this have more hydrogen bonding possibilities than regular proline? Yeah, it does. So, where do you see this? You see it in the cell walls of plants. Plant cell wall specific proteins have 4-hydroxyproline. You also see it in collagen. And the enzyme that hydroxylates proline has a cofactor called vitamin C. Vitamin C. And one of the symptoms of vitamin C deficiency is loose teeth, loose joints. We call that scurvy. Scurvy. And it was discovered that a little bit of lime juice, Captain Cook discovered that, a little bit of lime juice could stave away scurvy. That's why British sailors were called limeys, because they took lime juice, a teaspoon of lime juice, for every trip. And they still call them limeys. Hydroxylysine is also found in collagen. Why hydroxylysine? 1, 2, 3, 4, 5. 5-hydroxylysine. Hydroxylysine will be used to covalently bond to another lysine. That takes collagen fibers, links them together, makes them very tough and strong. In muscle, you have a 6-methyl lysine. 6-methyl lysine. That gets rid of the charge. Why might you want to get rid of the charge? Because you want hydrophobic interactions. That will allow protein to twist around itself, like in the tail of myosin, which is precisely what you want. And then when the going gets weird, the weird turn pro, look at this monstrosity. This is desmosin. Desmosin. So here we have lysine residues that have been merged into a ring. Merged into a ring. 3 lysines, all merged into a ring. This derivative of lysine is found in a protein called elastin. Elastin is found in stretchy sorts of tissues. Those stretchy sorts of tissues stretch and then stretch back, like your aorta. Structures like that. Desmosin is part of the reason they're able to do that. And you have gamma carboxyglutamate. This is found in blood clotting factors. Gamma carboxyglutamate is made by an enzyme that uses vitamin K as its cofactor. And this modification helps glutamate bind calcium. This makes it a very good calcium binder. Yeah. Sorry?
Which one uses vitamin C as a cofactor?
The enzyme so-called prolyl hydroxylase uses vitamin C as a cofactor. So you don't hydroxylate proline, you don't have vitamin C, you don't make good collagen. Don't make good collagen, you get loose teeth, you get retinal detachment, you get joint aches, get all the things associated with speed. Now, there are also other amino acids, but they are very rare. So selenocysteine has selenium in it. So this looks like cysteine, except instead of a sulfur, you have selenium. Selenocysteine is made actually from serine, and it is used in bacteria. It's also used in our cells too. We don't need very much of it, but this is the reason why you need trace amounts of selenium in your diet. This is amino acid 21. Here's essentially how you make it. So you take selenium and you activate it with ATP. There's a selenophosphate synthetase, makes selenophosphate. Then you take your serine and you attach it to a special serine tRNA to make this amino acylated tRNA. And this amino acylated tRNA has a serine, this special tRNA, the so-called sec tRNA, now is subjected to selenocysteine synthetase that uses pyridoxal phosphate as a cofactor. This activated selenium now replaces this group here with the selenium. And so this is made while it's attached to the tRNA. So this will get inserted at serine codons. It's inserted at serine codons. And it's only cells that make that special tRNA and that have these enzymes that are able to put that amino acid in. This is also very highly regulated. Don't want to make too much of this because you don't need very much of this. Amanda? Yeah, pre-translational, exactly. What sort of amino acids? What sort of proteins use selenocysteine? In prokaryotes, formate dehydrogenase and hydrogenase are two enzymes. Hydrogenase takes two protons and uses them to make hydrogen gas. Formate dehydrogenase converts formate to hydrogen gas and carbon dioxide. And glutathione peroxidase, thioredoxin reductase, and tetrahydrothiothionine 5'-deodorinase, which is a thyroid enzyme. So this enzyme is required in your thyroid, uses selenocysteine. And there is a 22nd amino acid. It was discovered in 2002 at Ohio State University. It was discovered in the enzyme methylamine methyltransferase from this very oddball Archaebacterium Methanosarcinum barkeri, and this amino acid is called pyrrolysine. Pyrrolysine. It is rare, very rare, and it is made also pre-translationally just like selenocysteine. And this one you don't need to know the structure of, just that it exists. Are there amino acids that don't find their way into proteins? The answer is yes. There are some that are metabolic intermediates. So this one, ornithine. Ornithine is a very important member of this cycle called the urea cycle. We won't study the urea cycle this semester, we won't have time. Those of you who stay with me for another semester, you will have to know the urea cycle. Then there's citrulline. Citrulline is a breakdown product of arginine because arginine is used to make nitric oxide. You convert nitric oxide, arginine, into citrulline when you make nitric oxide from arginine. So this is also not found in proteins. So amino acids are subject to acid-base chemistry. And if you dissolve it in water, you've got two groups here that are subject to acid-base chemistry, the amino group and the carboxyl group. The carboxyl group pKa is pretty low, 2 to 3. This is relatively high, high 8s, low to mid 9s, couple near 10. And this form is called a Zwitter ionic form. Zwitter ionic form, both of these are ionides. The Zwitter ionic form. So the Zwitter ionic form can potentially act as proton donors, acids. I can give this up. And they can also act as proton acceptors. They can act as bases as well. So here they're a sink for the proton, is where before they're giving. They're a source essentially of the protons. Compounds that have this ability to act as either proton donors or proton acceptors are collectively known as amphiolites. Amphiolites. And amphiolites usually have two groups. They are found in a zwitterionic form, and they can accept or donate protons. Because they have this characteristic of being both proton donors and acceptors, they become known as amphoteric. Amphoteric. One part of the molecule is a little base, the other is a little acid, so they're amphoteric. They kind of have the best of both worlds, shall we say. Well, amino acid would be one, yeah. That's the simplest example. There are others commercially that are used for 2D gels. Methylamino acid, methylamino carboxyl acid. That's one, and then there's others whose names aren't quite rolling off my tongue right now. So here we have examples of the pKa of the amino group of lysine, 2.34. pKa in the amino group, 9.6. Here's methylamine. Its pKa is about 10.6. Acetic acid, pKa is about 4.76, about 4.8, something like that. And these pKas are derived by doing titration curves. And if you do titration curves, you're going to get two different plateaus. And if you have a R group that has acid-base chemistry, you'll get three plateaus. You have three pKas that are derived. And the point at which, between the two pKas, that's a point where the amino acid is isoelectric. There's no net charge. That's the PI, the isoelectric point. So PI calculation, if you just have two groups, is easy. You add them together and you average them, and that's your PI. That's easy. It gets a little stickier. And this at this point here, 5.97 glycine is completely neutral, electrically speaking. And here's what the titration curve for glycine looks like. Again, this is not a guess. These are very well worked out. These numbers, as you can see, are very precise. They're known quite well. This is what these curves, if you've never done a curve like this, a plateau essentially, that's the spot at which you get pretty good buffering. Here, you won't get very good buffering, but the midpoint between this, that's your PI. Now, what happens if you have, yeah. Oh, you can see it. If you're nowadays, as the micro lab, it graphs as you go. You can see it in the graph. It's really obvious. Because first you get usually this, yeah, like that, and then all of a sudden it gets really flat. And then it goes, yeah, again, like that. And it's just pretty obvious. And the PI, you can eyeball it, but eyeballing is never very reliable. You let the computer calculate it for you, because that's always more accurate than your eyeball. And yeah, that's what these things look like. Pretty unmistakable. So if you get two groups now that have acid-base chemistry, calculating the PI gets a little more involved. In this case now, because these two are -1 and this is +1, that means you want a spot where this is -1/2 and this is -1/2. So you want that spot at which this is definitely +1, but these are each only half ionized, so it's going to be the point in between these two. So your PI will be 2.19, 2.2 plus 4.25. That's divided in half, and that's 6.4 something, so it's 3.2 something, whatever. And that's how you calculate that PI. And then if you use something with a positive charge on the R group, histidine, for example, it will be between these two. So this to be minus, plus, minus, plus 1/2, this to be plus 1/2. It'll be between these two. This will definitely be -1. So it'll be 6 plus 9.17, is that 15.17? So it's 7.5, 7.5 plus, 857 585, 7.59, something like that, or close to that, or thereabouts. And that's how you simply do the PIs of these amino acids. Okay, questions? Anything? No, screen sharing is stopped, of course, because I stopped it. No? Yes? No? Okay. Then let's take a look at peptides. We'll only be able to start this. Right. Oh, crumbs. Yes, enable editing. There. Thank you. Yes, enable content. I don't care. Stop being that way. Okay, let's share screen again. Okay. So what is a polymer of amino acids called? It's called a peptide. It's called a peptide. What's the difference between a peptide and a protein? Good question. There's a very arbitrary decision that's been made, and it's that if you are functional and you have a molecular weight greater than 10,000, then you're a protein. Is that arbitrary? Yeah. But that's essentially the way people define it. So amino acids are linked together by something called a peptide bond. Peptide bond. This bond is directional, very, very directional. And it's made between the carboxyl group of one amino acid and the amino group of the other. And essentially, what happens is that you get a you get the production of a who's this? Water. And then you form a bond essentially between the carboxyl of one and the amino group of the other. And that's your peptide bond essentially. Very, very directional bond. So you always have an amino acid in the front, you'll always have an amino acid in the back. Amino acid in the front will have a free amino group, the one in the back will have a free carboxyl group. Highly, highly directional. It's made by means of a condensation reaction. All right, now that's a good place to start. Don't forget to clean up. And I will see you guys tomorrow. We're going to do the carbohydrate lab tomorrow. And you'll work in pairs. You'll have an unknown. And then you'll figure out what your unknown is doing the test on it. Okay? All right. See you tomorrow.